remember that we are discussing the Kobasa et al. Paper on Monday. And you need to turn in the answers to the questions about that paper at the beginning of class on Monday. The paper as well as the questions are on Moodle. So just be aware of that. And today I'm going to continue talking about the cellular part of the innate immune response. This is always a really tricky lecture for me because there are a lot of parts that are related to my research. And so I want to go on and go into all sorts of details and get way excited about things. So I'm holding myself back. You will also notice I have more slides than I will possibly get through today, but I like them all, so I couldn't possibly get rid of any of them. So I put them there with the knowledge that I would probably not get through all of them. We'll see what happens. There are a few different things I could imagine happening on Monday if there's time left after the paper. One of them could be more slides. There are also other things, so we'll see. But here is sort of that idea about the cellular innate immune response. And this is what we started out talking about on this, or we left off with on Wednesday. So we've been talking about this process of inflammation. We saw sort of an original body site, in this case, a site in the skin that was sort of in a normal situation. Then we saw infection happening in that situation. And we talked about the macrophages that live in the tissue, recognizing the microbes, using the pattern recognition receptors that I mentioned to you last time, and then making this new product of an inflammatory cytokine. They make that product by transcription and then, of course, translation and secretion. A lot of times people measure mRNAs, measure transcripts here, and I point that out because the paper that we're going to talk about on Monday has some parts that are really key about measuring transcript. And we make these inflammatory cytokines. And where we sort of left off was thinking about the details of those inflammatory cytokines and chemokines in terms of exactly what they do. So there are many different cytokines and chemokines that are produced here. The two that I mentioned by name were TNF alpha and IL-6. And they are two who, no, stop. Come on. They're two that you will hear about again later in the semester. So you should kind of recognize TNF alpha and IL-6 as two of the big inflammatory cytokines. Another one that is really important here that's not on this slide is called IL-1 beta. And so sometimes IL-1 beta, TNF alpha, IL-6 are sort of made by these macrophages to induce inflammation. For your purposes, I'm kind of going to lump those three together as the three of them are happening together and doing similar kinds of stuff. Again, later in the semester, they're going to show up again. So I wanted to highlight them here. And you can see that these cytokines, as well as some of the others that are made, will lead to a number of different changes in the body in order to lead to this state of inflammation to block replication of microbes or to clear them, to somehow end this infection problem. And when we think about the effects of these inflammatory cytokines, we can very broadly divide their effects into two different groups. One of those types of effects are the local effects. And so the information that you're going to see here, as well as sort of like on the next slide, will be local effects. And then we're going to see another type of effect on subsequent slides. So the local effects are the effects in that specific area. So if we want to think about an idea of local effects, Again, remember there's this example that everyone uses about you pound your hand on the podium while you're giving a lecture and you get a cut on your hand. The local effects are things that are happening in your hand or things that are happening right near that cut. And they're not happening elsewhere in the body. It's just at that specific location. So those cytokines are being made, and one of the really important local effects, well, actually two of the important local effects are changes to the blood vessels and that specific location. So these changes aren't happening to all the blood vessels in the whole body. They're just happening to blood vessels really close by. And you can see that one change is we see a big change in the diameter of the blood vessel. So it goes from being this big to being this big. That's called vasodilation. We also see that the cells of the blood vessel wall get less close together, so the blood vessel gets leaky. That's called an increase in vascular permeability. And so you can see both of those changes happening here. That means that more liquid from the blood can go into that site, and that liquid contains all sorts of proteins, like the antimicrobial peptides and complement we talked about before. So those could stop bacteria in this location. This also makes it possible for cells to leave the blood vessel and go into that site. And so we get fluid, protein, cells leave the blood, enter the tissue. This, of course, makes the tissue have to get bigger to accommodate all of this stuff. So we get some swelling. We get some redness from this blood coming in. We get warmth from the warmth of that warm blood coming in, as well as some other metabolic changes. And those, of course, are 3 of the 4 cardinal signs of inflammation. Those inflammatory cytokines do also act on the nerves in order to lead to pain, the 4th sign of local inflammation, though I'm not going to talk a ton about that because that would require us to talk more neuroscience details. But note that evolutionarily, if you have a wound, it's probably a good idea for you to protect it and not like bang it on the ground and let it get worse. And so having pain, so you're like, oh, look, I should do something about this, is actually pretty important evolutionarily. One other thing to mention is that last time when we were talking about complement and the ways that complement could act against microbes, we saw 3 different big effects of complement. And one of those effects I told you about was inflammation. Oh my gosh. And when I told you about inflammation, I told you about specifically the complement fragments C3a and C5a. And I said that they can float off and lead to inflammation. It turns out that the C3a and C5a receptors are basically identical to chemokine receptors. And for all intents and purposes, the things that C3a and the things that C5a do are change the size of the blood vessel and make the blood vessel leaky, like increasing vascular permeability. And for almost all intents and purposes, as far as you care about, C3a and C5a act the same as inflammatory cytokines. So this is another place where kind of different things that we've categorized in the immune system are showing some similarities. So yeah, technically, these were part of proteins of the innate immune system. And the cytokines we're talking about are part of cells of the innate immune system, but we're seeing the same kind of themes and features going on. And so many of the effects of C3a and C5a are actually really similar to these effects of inflammatory cytokines. And one other really important effect, and this is true of C3a and C5a, it's also true of some of the products made by that macrophage as the result of the signaling, is that we can actually have the area specifically recruiting other immune cells. So some of the signals that are sent out are signals that actually attract other cells. So it's not just that the vessel gets leaky and big. We also actively are calling and attracting other cells and saying, hey cells, you need to come here. So we will see active recruitment of other immune cells at this point. Those other immune cells will be recruited by a specific process with a number of steps. That's shown in figure 3.24 from your textbook. Those steps that are shown here are actually steps that immune cells use to go to all sorts of different parts of your body. I'm not going to focus on the steps right now because they're the same steps that happen in a bunch of other places, and I like to put them in a different place in the immune system. So we're going to see those steps for T cells to get to a place, but it's the same steps. So if you happen to, so you'll probably see this figure again, and if you kind of looked at that figure and we're trying to get information about it, we're going to see it with T cells, but it's the same thing. Sometimes this is due to chemokine activity. So remember, chemokines are cytokines that make cells move. And so this can be due to production, particularly of some chemokines. As a result, we end up with a situation like this. And so this is my overview of local inflammation. We have our person who was banging their hand on the podium. They got a cut in their hand, and they had that inflammatory response happening in their hand. And you can see it's happening in their hand because their hand's red. And so we see changes in the blood vessels just there in their hand. So we see, more phagocytes, migration into the tissue, changes in the blood vessel wall. We see leakiness of the blood vessel, all the things I just showed you. But notice the rest of the body is not shown as red. The blood vessels everywhere else in the body are not changing. They're staying exactly the same. We're just having this inflammatory response in this one location. And then that sort of contains the infection to that one location and cleans it up. And then the person lives happily ever after. See, they survive. So we've cleaned up the inflammation, we clean up the bacteria, the inflammation stops, we go back to happily ever after. This is kind of what we think about for local inflammation. There are also some effects of those cytokines in other parts of the body. And this might happen because those cytokines could get into the bloodstream and maybe travel to sites further away. And so these effects are known as the systemic effects. So these are effects that are happening in other parts of the body outside of that hand that is undergoing inflammation. Some of this is based on things like how much cytokine you have. Maybe if you only have a tiny bit, it'll just stay in your hand and not really go many other places. But if you start to have more made, you're going to start to see them go through the bloodstream and impact other tissues. One of the other tissues that will be impacted will be the liver. The liver is going to make some new proteins and secrete those proteins. One protein that you see here is called acute phase proteins. You don't need to know a whole list of proteins here, but I'm going to give you some examples. Some examples include all the complement proteins, as well as a lot of blood clotting proteins. So when we start to have inflammation, you can see it's IL-1 beta, IL-6, TNF alpha, the three I called by name earlier. We suddenly say, hey liver, can you please make some more complement? Because we're doing a response and we're probably going to use up our complement. We should have more. So again, these two different responses that we've talked about are tying together. These cytokines will act on the bone to say, hey bone, release more neutrophils from the bone marrow. When we talked about cells of the immune system, I told you that there are a whole bunch of neutrophils that hang out in the bone marrow that can be released if we need more. It's like the SWAT team that we can just release from the bone marrow if we need more. So we see the signals going to the bone marrow and saying, hey, release more neutrophils for us. We also can see some changes in the brain and changes in fat and muscles, all of which are really sort of metabolic types of changes or changes that influence body temperature. So we can increase body temperature that increased body temperature stops microbes from replicating, because bacteria and viruses often have kind of a best replication temperature. So if we just raise the body temperature, those bacteria aren't going to reproduce as well. So it kind of slows down those numbers issues that we talked about before. We also have learned that some of the metabolism parts of this are important. And we've also learned that the increased body temperature is actually makes some of the white blood cells work better, and it makes them travel to sites better. Both of those things are things we've actually learned more in recent years than before. If you were going to tell someone about this effect, let's imagine this effect was happening to you. How would you describe it to your friend who's not taking this class? What happened? Yeah, Michaela. I have a fever. This is a fever, is all this is. So what you should notice here is that a fever is actually not something that a microbe did to you. A fever is something that your immune system did to your body to try to improve aspects of defense. So here you can just see, I'm showing you sort of zoomed in examples of some of this stuff. So here are those cytokines acting on the liver, making lots of different proteins that are involved in things like complement and blood clotting. It's exactly what I said to you before. Yeah, Josh.
So at which point does this go wrong and the fever gets like crazy?
So Josh asked the question, and there are a bunch of, I was sort of, it's a question that comes up here often, or at least versions of that question come up here often. Josh asks about sort of where the line is between the fever being bad and the fever being good. How else could you imagine asking that question, or sort of what other things does that kind of tie into? Because like I said, there are a few different ways to think about this.
How I imagine it's since we're talking about the side effects, it could be probably increase, like your body's just increasing the amount of inflammatory cytokines, so that response is going to be stronger in a way.
Yeah, so part of this has to do with how much inflammatory cytokine you have, and that leads to how high the temperature is and all of that stuff. So microbes have a specific temperature they like to reproduce at. So we're part of what this is going on here is we're increasing the temperature to a point where they don't reproduce so well. Your cells don't love those high temperatures either. And so if you go for too long at a high temperature, it's going to start being detrimental to your cells too. And so part of it is about the length of time. Part of it's also about how high the fever is. And some of the really high temperatures can be problematic for you physiologically. But the other place where this, sort of the other way that people sometimes phrase questions about this is, does that mean it's a bad idea to take medicine to get rid of a fever? Is basically kind of a similar question of like, does that mean if a fever is something my immune system is doing, does that mean I should like keep the fever and not take a medicine to make my fevers go away? Am I hurting my immune system by taking ibuprofen? It becomes a question that people ask here. And there are different studies about the details of it, and sort of different immunologists will pick and choose at different details of it. I'll tell you my personal answer is that it's about whether the fever is impeding me from like carrying out my daily activity. So if I, have a fever, but it was a Saturday and I was planning on watching Netflix anyway, I just like stick with having a fever and sit on my couch and watch Netflix. If like it's impeding some aspect of my daily activity, I might take something. But I try to avoid it if I can, is my personal answer to that. And there are papers that sort of argue different pieces of this. But that question is also tied to the question that you asked. So yeah, here's the acute phase response, one of the responses. Here's the bone marrow response, one of those responses. So it's just showing you in picture form, oh my gosh, there's all the neutrophils in the bone marrow, and we're going to release them because of those cytokines. And then this is that same thing again to tell you about body temperature increase. And so this is just another way of thinking about this. In larger quantities, you're getting those cytokines in circulation, and they can have a bunch of systemic or global effects in the body. On the bone marrow, on the liver, on the brain. Some of those other effects include things like making you sleepy, making you kind of lethargic, making you have no appetite, making you nauseous. All the things you think of as being sick are actually directly coming from those inflammatory cytokines. Some of it may have to do with changing how your body's using energy. Like, okay, we're going to give all our ATPs to defense, and we're not going to stay awake. We're going to take a nap and let the body focus its nutrients towards defense. And so pretty much a lot of the things that you think of as sort of sickness, as feeling bad, are actually because your body has inflammatory cytokines being made in it. Sometimes when people will tell me about getting vaccine doses, they'll be like, oh my gosh, I feel terrible after getting a vaccine. I have a fever. It's so bad. And I'm like, yeah, that's because your immune system is like responding. Like your immune system is making a response and doing what it's supposed to. Yay. And so when my friends post on Facebook like about how bad they feel after a vaccine, I usually write some comment like, I'm so happy to hear your immune system is working as it should. And no one's ever punched me yet. But notice that some of these effects that people think of as really sort of the bad things are in reality adaptive things that your immune system is doing. And once we have our inflammatory cytokines produced by the macrophage that was originally in the site, we can now have cells coming into the site. So our cells have come to the site. We may also have had proteins come to the site, other things. And now we finally have to actually get rid of those bacteria to finish this whole process. Technically, we also have to, like heal the wound and clot the blood. But immunologists classically haven't concerned ourselves with that, and we're only now realizing that was maybe dumb of us. But that's okay. And so the final piece that we have to think about is what goes on for those cells that come in to actually kill the invading bacteria. Remember at the beginning of this process, the cell that was in the tissue that started this whole thing off was a macrophage. And remember, it could do phagocytosis and kill microbes. It also had pattern recognition receptors that could allow it to eventually transcribe cytokines. The cells that we're going to see acting here at the end are a different cell type. So we're not going to be seeing macrophages here. Here, we're really seeing the actions of neutrophils. And so now those cells that come in are neutrophils. So we're still in innate immune cells, but we're in a different type of innate immune cell. I will fully apologize right now. I spend a lot of my time thinking about macrophages, and I still probably, to my detriment, have in my head a little bit that like neutrophils are like the dumb ones and macrophages are the smart ones that do fancy things like PRRs. Neutrophils do have some fancy things they do too, but they don't do that whole PRR signaling and making cytokines. And that's the thing I think about a lot. So I'm like, neutrophils. One thing that neutrophils and macrophages have in common is that they both can do phagocytosis of invading microbes. So our neutrophil can bind to a microbe, and it has a lot of different types of receptors that can allow for that binding. Now we have done phagocytosis and we make a new organelle, which is the phagosome, because it's the organelle that got made by phagocytosis. The phagosome will eventually fuse with the lysosome. You can see that the pH is going down here. So we're getting more and more acidic. And that acid, as well as a bunch of other enzymes, will allow for complete degradation of that microbe. When we talked about neutrophils earlier, we talked about them as granulocytes. I mentioned that they were granulocytes because they had a lot of little granules in their cytoplasm. You can see it, you can sort of see the little dots in this image. This picture shows some granules around the cell. And the idea with those granules is that they are full of different kinds of enzymes to kill bacteria, and those granules are all fusing with this phagosome to just deliver these toxic enzymes to help the process. So that's what the granules are doing. Yep.
Wait, so you said earlier that the granules in the cells, like they shoot them like toxic bombs?
So that's basophils and eosinophils. Okay. Neutrophils instead shoot them more like toxic bombs inside the cell. They shoot them as a toxic bomb towards the phagosome instead of out. Okay. Yeah. So basophils and eosinophils are shooting them out. Here they're fusing with the phagosome to kill things that are in the phagosome. Yeah, absolutely. Because I would feel remiss if I didn't do this. This is a video. It's on YouTube. You can find it everywhere. When I was just looking for it, I didn't know they had this one, but apparently they have it set to the Jaws theme too. And we're going to watch this neutrophil, which is right here, dealing with some Staph aureus bacteria, which will be these little dots. These are red blood cells, which are contaminants when they did the experiment, but they end up looking like fun obstacles. And so we can see what's going to happen. So Andrew asked a good question, which is, why doesn't the phagocytosis happen immediately? I really like that question because that means that I can highlight something that we see on this slide. One thing to note, when you watch that macrophage doing phagocytosis, you actually look very closely, it did change its cell shape quite a bit, right? There were a lot of, in order to actually eat, it had to extend parts of its membrane, it had to then move parts of its membrane. There's actually a lot going on with actin in terms of phagocytosis happening. And so that cell actually does need to be doing a lot of cell biological changes. It isn't able to move immediately in order to come in contact with that microbe and engulf it. And so there are a lot of cell biological changes that have to happen. One reason why I think it's important to point that out is that a lot of people will sometimes think about if a cell takes anything inside it, they call that phagocytosis. But phagocytosis is actually a pretty regulated process involving a lot of things like these actin rearrangements. It turns out your body really only has, I'm trying to think if I'm being wrong here. Your body has 3 types of cells in your whole body that know how to do phagocytosis. And that's actually neutrophils, macrophages, and dendritic cells are the only ones that can do the real phagocytosis. There are some other ways cells can internalize stuff, but they don't have as much effort involved as phagocytosis, so they actually don't qualify, and we'll see them later. Sometimes people in other classes will be like, such and such happened by phagocytosis, and I'm like, actually that's not the process in here, but I try not to be too annoying. Yeah, Josh.
So does neutrophil just kind of push their blood cells out the way? It's get out of the way.
Yeah, it's really just completely pushing them out of the way. I mean, it's done in a dish, so it's a little artificial, but it's fun to watch. But then the other piece of this we can think about is sort of what's happening at the end. And so we basically are going to digest that microbe to the point of it being things like constituent amino acids and things like that. And so you can imagine the cell might want to recycle that stuff and use it for its own biochemical purposes. More and more, what we really understand happens actually with this is something pretty important, which is that neutrophil and all neutrophils, if you go back to my table I showed you in the cells and organs lecture, you would see that neutrophils have the shortest lifespan of any of the white blood cells. And so after a neutrophil eats microbes and sort of starts to do this process, it dies. Neutrophils are very, very, very short-lived. And so you can see that our neutrophil will engulf a microbe. You can see that we'll have our granules and they will fuse. Here you can see all our granules fusing. You can see our degradation of this bacteria. I realize I'm doing this slide in a different order than I thought, so we're going to get back to that death in just a second. I have one other thing I want to tell you about the degradation, which has to do a little bit with some of the things that are involved in the degradation. Some of those things are just enzymes, which hydrolyze things. And so this part of the slide says hydrolases. There are also a number of different compounds that are made just inside of the phagosome and the lysosome. So the cell's making these compounds, but it doesn't like let the compounds go everywhere, float around the cells. It holds them just in this little compartment. Some of these are types or versions of oxygen, either the superoxide anion, the hydroxyl radical, hydrogen peroxide, or hypochlorous acid. And you can see them here. There are enzymes like superoxide dismutase that will make some of these things. And so just in that little compartment, we'll make some of those. We also can make some things that involve nitrogen, like nitric oxide, nitrogen dioxide, and peroxynitrite. And you can see the details of this here. The process of making these compounds inside that phagosome is called respiratory burst. And so sometimes we say that the way that we can destroy microbes in a neutrophil is by respiratory burst, because we're making these different compounds. Now I want you to look at some of the oxygen versions. We often refer to them as the reactive oxygen species, or ROS. So we'll look at the ROSs for a second. This third one here. Do you know this chemical at all? Some people are shaking their head yes. Tell me what you know about it. Yeah, Kyra. So if you put it on an open wound, it gets all foamy. Okay. Why'd you put it on the open wound? Hey, that's the thing you put on your skin to kill bacteria, isn't it? Does it kind of burn a little bit when you put it on? Because it's not great for your skin either. It's kind of toxic for your skin, but it kills bacteria. Hypochlorous acid. This is the active ingredient of bleach. So basically, in this little compartment, your cell is making bleach and killing the microbes and making peroxide. If it went everywhere in the cell, that would kill the whole cell. It would kill your cell too. But instead, we just make it in this little compartment. We make all these chemicals, and then we're basically just like bleaching the microbes inside a compartment in our cell. So none of this is like rocket science here. There also are a number of different antimicrobial peptides, like some of the antimicrobial peptides we talked about before, that happen to be in those compartments. So again, the proteins that we saw before do play into what we're seeing with the cells of the innate immune system, because things like lysozyme will be in some of those compartments, and other antimicrobial peptides as well. Now, finally, the neutrophil dies. This is where I forgot that I only was showing you part of the slide and not showing you the end. So here is really kind of what happens with a neutrophil. That neutrophil will do phagocytosis. It will degrade that microorganism due to enzymes, due to respiratory bursts, due to antimicrobial peptides, due to the acidity, all that stuff will help us degrade it. But then that neutrophil is going to die by apoptosis. That neutrophil is very, very, very short-lived. And what will eventually happen is that some macrophage will come and eat the neutrophil's body. We'll just come eat that neutrophil. So you can see here's a macrophage. It is phagocytosing the neutrophil. And that way the macrophage is getting all of those amino acids and nucleic acids and lipids and carbohydrates, all of those things that we've got here. So basically, like, we're recycling the neutrophil and we're recycling all the parts of the microbe. You actually know this, even though you didn't know you knew it. Because there is oftentimes when you're seeing a site that will be finishing in a process of inflammation, that body site will have a lot of dead neutrophils. What color, this is a really hard question. What color are white blood cells? white, right? So what color do you think dead neutrophils are? White. You know how sometimes when you have like a cut and it's healing and there's like that white goop all over it? That's just the dead neutrophils. It's called pus. Pus is just a collection of dead neutrophils that are made at the end of one of these responses. So you actually knew that all along. You just didn't know what was going on there. I also will tell you, as I have learned from experience, sometimes in situations like this, I like to put images on slides. And so when I was making this slide, years ago when I was making the slide, I did a Google image search for pus so I could show you some pus. And what I learned is that if you do a Google image search for pus, you see a lot of things you did not want to see. And so that's why it just says pus there. All right. So the neutrophil dies. And this is kind of the original discussion of the neutrophil. In more recent years, we have learned that the neutrophil can do one other thing. And this is the neutrophil's special function. Just like with the macrophage, it had that special function of making inflammatory cytokines. We have found a special function for neutrophils. And I will admit, it's actually kind of cool. And that special function actually has to do with the way the neutrophil dies. When that neutrophil dies, the neutrophil actually expels some of its internal components. And specifically the thing that is expelled is the histones and the chromatin. So the DNA and the chromatin get expelled. So the neutrophil just throws all its DNA out of the cell. It turns out that DNA and chromatin mix is kind of sticky. And it forms a trap where other bacteria outside of cells can get trapped. Honestly, people love to joke now in the field about how neutrophils are basically Spider-Man. They shoot this like sticky web out of their cell as they're dying. So if there are any bacteria in the area, they could get stuck in this web of DNA. This is known as the neutrophil extracellular trap, or a neutrophil. Which net is what that stands for. And so we often call these neutrophil nets. And sometimes we talk about the process of dying and throwing a net as netosis. And so these are figures from your textbook of bacteria caught in the DNA nets that have been expelled from neutrophils. So these are a bunch of E. coli, and this stringy stuff is the DNA net that was expelled from a neutrophil. And so now you can see all these are trapped. They're not going to go infect other cells. And here you can see, again, you can see cells and this sort of matrix around them of neutrophil net. And this is coming from your textbook. So that was my very broad discussion of some aspects of inflammation. As I mentioned to you last time, inflammation has a bad reputation. Oftentimes people talk about wanting to get rid of inflammation, all the problems that happen with inflammation. And it is true that I can link so many diseases you wouldn't believe it to inflammation. And then I say they all happen because of immunology. And in fact, they all happen because of PRRs, which is what I study in my lab. And so I study all the diseases because they all have inflammation as part of them. And so we can also think, and I reminded you last time that inflammation has a really important physiological purpose of defense, tissue repair, restoration of homeostasis. If we had that person who slammed their hand on the podium, gets the inflammation in their hand, they didn't have inflammation hit there, they would have like bacteria reproducing out of control and disaster would strike. Inflammation can also go wrong. And this is sort of implied by Josh's question earlier and just implied by a lot of different things. And so if it goes wrong, we can start to have things like autoimmunity or other kinds of inflammatory diseases. We can start to have other types of tissue damage. We can have changes to homeostasis in the body. And so I want to say a couple of things about kind of why inflammation can be bad as much as we've talked about why inflammation can be good. One of the most famous issues related to inflammation is something called septic shock. Many patients in hospitals, particularly patients who go to a hospital with bacterial infection, will die of septic shock. In fact, the 2011 Nobel Prize was given for some understanding in innate immunity, and one of the parts of it was actually because they're like, this could help us save a lot of people who have septic shock. And so septic shock's one way this can go wrong. And what happens with septic shock is that we will have some kind of trigger that induces inflammation, not in one area of the body, like just the hand, but everywhere in the body at the same time. So this figure is saying that this is some kind of bacteria in the blood. So you have this bacteria in the blood, then you're going to have inflammation in the hand and also in the head and also everywhere else. So you can see the whole body's red. Let's think about what happens in that situation. Remember our example with the hand. What happened to blood vessels in the hand? They dilated. What else happened to them? They got leaky, right? That's one thing that happens in your hand. In your whole body, every blood vessel in your body expands and every blood vessel in your body leaks. What's going to happen to you? What? You might swell up, okay? So you're going to swell up because you're going to get fluing in weird places. You will notice this says systemic edema. That means swelling. So you're going to swell up. What else might happen? So if all your blood leaks out of your vessels, what's in your vessels now? Nothing. So suddenly you have no blood anymore. You've lost blood volume. Your blood vessels might actually collapse because there's nothing in them. You're going to not have enough protein going around the body. You're not going to have enough cells going around the body. So basically you're going to actually end up with like not enough nutrients and things moving because you got no blood in your blood vessels because it all leaked out. And like I said, collapse of the blood vessels. You also often will start to have all sorts of clotting like everywhere in your body, which will block vessels. And eventually this leads to multiple organ failure, and the patient will die. So local inflammation, a little bit of inflammation, a short time in one period, in one place of your body is good. If you had your whole body doing this at the same time, that is disastrous. So that's bad. The other way that inflammation can be bad is if it's not necessarily happening in your whole body, but if it starts to happen for way too long. So you start making those inflammatory cytokines, and you make those inflammatory cytokines for not a day or two, but for like a year or longer. Remember, one of the effects that we saw was a change to muscles, right? And we saw that change to muscles in order to change metabolism, to help with fever. Well, eventually, if you hit that muscle for too long and change the metabolism of that muscle, you're going to like permanently change the metabolism of that muscle. You may end up leading to insulin resistance in that muscle, which is part of type 2 diabetes. You might, if that muscle is the heart, influence heart output because you've now messed with the heart and its ability to beat well. And so suddenly you might end up with cardiovascular disease. You might actually change the blood vessels because the vessels are getting hit so often to the point where you might get other cardiovascular issues because of changes in your vessel's permeability or because of clotting. And so the other problem with inflammation is if inflammation goes on for too long and you start to have some of those physiological changes be happening for longer periods of time. And so one of the big things that a lot of immunologists spend time thinking about is, why might you have inflammation going on too long? We seem to have a bunch of these different diseases, particularly in sort of modern times. That might tell us, well, I guess people must be having inflammation for longer periods of time than they did before. Like, why is that? We don't know. We have some hypotheses, though. And so we have these ideas that there may be situations where people are undergoing chronic inflammation for super long time, maybe like the length of their life, or years. And what we can see is that chronic inflammation will start to lead to things like death of different cells in some tissues, will lead to scarring as part of that wound healing response, will lead to weird blood vessel growth, will lead to those metabolic changes, might lead to obesity, and eventually could influence things like give people type 2 diabetes, heart disease, atherosclerosis, organ failure, cancer, Alzheimer's, autoimmune disease, autoinflammatory disease, inflammatory bowel disease, asthma, transplant rejection. I could go on and on and on. Basically, almost a lot of diseases can be tied back to chronic inflammation. Some of this we think is due to genetics. Some people seem more prone to chronic inflammation than others. Some of this also we think is related to things like maybe we're making weird responses to the microbes in our gut, or we have infections that we didn't know about that are pushing inflammation for long periods of time. We're also realizing that there are some environmental factors that seem to trigger PRRs to lead to inflammation. There are actually some lipids that turn on certain PRRs. So in fact, maybe some dietary lipids that are also associated with obesity are giving us a lot of inflammation. There's a really cool paper about feeding mice different lipids and the changes in inflammation. Maybe it's other types of things that we're seeing in our life. There's some things about like certain environmental pollutants that we're breathing in or whatever. But all of those things could be hitting PRRs, leading to chronic inflammation and pushing the development of a lot of these different disease states. Yep.
Is there any indication of like stress activating chronic inflammation?
Maybe. So right before you said that, or right as you were raising your hand, I was thinking in my head, there's this one other issue that I haven't mentioned, and it's a little bit of a rabbit hole, and it's interesting, and I have to decide if I'm going to mention it or not, and it ties sort of into your question. So I'm going to ask you guys a question, and I promise this will come together, okay? So, there are a bunch of situations where you probably have had inflammation happen in your life, right? And some of them are like you get the cut on your hand, right? Can you think of any other times when you might have had inflammation? Once when I was teaching this class, I actually had this issue happen to me the day before, and so I got to have a demo in class of this inflammation. I'm really clumsy. Really, really, really clumsy. What could happen to me if I'm really clumsy? Yeah. I could fall. I could trip on something, and what might happen to me if I fall? I get a bump, or like I twist my ankle, right? And in fact, I messed up my ankle real bad right before class one time. And it was literally, and so I twisted my ankle. Imagine a twisted ankle. It gets super swollen, super red. All the things we just saw happening in inflammation, right? It was inflammation. Here's the problem immunologists struggle with in that situation. What the heck turned on the PRR? My skin didn't break when I twisted my ankle. No microbe got into my ankle. How did the PRR get a signal to make those cytokines? So this is a situation known as sterile inflammation. What we think happens in sterile inflammation, though this is a somewhat controversial area, is that we think sometimes when cells die or when cells are damaged, they leak products that start inflammation. So I told you about MAMPs, or microbe-associated molecular patterns, but there's also this idea in the field that there are things called DAMPs, or damage-associated molecular patterns, that are made when cells are damaged that may also lead to an inflammatory response. And the most obvious example there is my twisted ankle. So I could imagine under cases of stress, Emma, that you could see cell death or cells just undergoing damage and releasing damps in the same way. But because the whole damp thing is not as well understood, it's sort of hard to fully work that out. Does that make sense? Okay. All right. So here's our overall inflammation process. One piece that I want to point out about this process, as I've shown it to you here, is that it involves, in this image, some bacteria. And the things that I'm telling you about in this aspect of telling you about inflammation are sort of parts of a classic response to bacteria. We also understand a similar response that happens as a result of certain viruses. Where we draw the line between inflammation and this virus response, it's tricky. So I'm not going to often I think about the inflammation as the bacteria thing, and I'm going to talk about I think about this virus thing as different, but that's because I study one versus the other. So I don't think most immunologists separate them in their mind quite as much as I do. But we need to talk a little bit about what goes on with viruses. So here you see two different images of a virus infectious cycle. So viruses infecting a cell. What I want you to notice in both of them is that the virus will interact with some aspect of the cell's membrane, some receptor on the membrane, and then come into the cell and uncoat to the point that we just have nucleic acid. So here that all kind of happened by magic. Here it's shown in some more detail. And then we've got this nucleic acid. It could be whatever nucleic acid, RNA or DNA, depends on the virus. We don't care. We just care that it's nucleic acid. And eventually we're going to make some new proteins and take those proteins, assemble them together, make new progeny viruses, make new baby virus. If we look at this, there are a couple of problems with what we've been thinking about with inflammation. And the biggest problem is actually a huge piece of virology is how viruses do or don't get into a cell. Does the virus have the ability to get into the cell? So if you have this virus hanging out right here and you do phagocytosis, is that going to be the best way to deal with that virus? Not necessarily, because you just let the virus into a cell. That was kind of the virus's whole sneaky goal anyway, and you just let it happen. So phagocytosis isn't always a great way to get rid of viruses. And if you were to think about time here on this slide, most of the time of the virus's life, it's inside one of your cells. Can you phagocytose one of your own cells? No, that doesn't, size-wise, that doesn't work. That's just bad. There's really no good way of making that work. So the process we just talked about is not going to be super, super useful with viruses. One other piece of this, and I could go on about this for a very long period of time, but I will not hold myself back, is that notice kind of one of the key things that happens with viruses is that we get down to their genome, their nucleic acid. When we get down to their genome, it's really hard to do anything about it. We don't have little tweezers that say, this RNA I don't like. You take it out of the cytoplasm and throw it away and leave all the other RNAs behind. Once it's just nucleic acid in the cytoplasm, it's nucleic acid in the cytoplasm. There's kind of nothing you can do about it. And that's something that you should understand in general when we're thinking about viral infection of cells. Once a cell is sort of doing this process, once a cell has a virus infecting it and is going through this cycle, that cell basically is going to become a virus production factory where it is going to make thousands or millions of copies of the virus. Yep, Andrew.
Couldn't they just possibly do apoptosis and just pop themselves?
So Andrew asked a great question. Couldn't they do apoptosis and pop themselves? And note that the idea there is the cell dies, right? The exact point that I was going for is I wanted to say, at this point, we really can't save the cell. At this point, there's nothing we can do for this cell. So either the cell has to kill itself, or we have to kill this cell, because we don't want it to be a virus production factory. So the exact point that sort of you're getting at here is right. The idea is, once we're at this point with the virus, we're not doing anything good for the cell. And so we're going to see this in a few different places throughout the semester of, we got the cell infected with a virus, we need to actually get rid of that cell. In the 1950s, some scientists were doing experiments with a virus, influenza virus, and putting it in cells in a dish. And what they found is that when a cell got infected, it made a magic product. It just made this cool product. And if you took the product that it made, so it made this product and it secreted it from the cell, kind of like a cytokine. It kind of is a cytokine, but they didn't know that then. They made this product that got secreted from the cell. So if you have your cells in a dish, they've got the liquid on top of them. You could just take their liquid and put it on other cells, and suddenly those other cells could not get infected. There was like magic infection prevention liquid that an infected cell made that prevented other cells from getting infected. They didn't know what it was. They didn't know any details of it. They just knew that this magic stuff interfered with infection. So they called it interferon, because it interfered with infection. We now know that there are a bunch of types of interferon. They're listed here as type 1, type 2, and type 3. You can see type 1 is interferon alpha and beta. Type 2 is called interferon gamma. Type 3 is called interferon lambda. And the ones that I want us to care about right now is type 1 interferon, or interferon alpha and beta. And those are also going to show up prominently in the paper we discuss on Monday. I will just tell you, interferon gamma is made later and is part of the adaptive immune system. So we will care about interferon gamma later. We're done with interferon gamma until adaptive immunity. Interferon lambda is known to be made early. It's a little bit newer. We're not going to talk a ton about interferon lambda or type 3 interferon. You might see it mentioned here or there in a few places, but we're not going to talk about it a ton. We're really going to talk about type 1 interferon, which is interferon alpha and beta. It's made as a result of viral infection. So you can see it says inducers, viral infection. And it is made really rapidly after viral infection. So within hours. It's made early, and it's by virus-infected cells. And in fact, it goes away pretty early too. So you can see there are some ideas that it's turned on and then starts to go away at 10 hours. So we're talking like right after viral infection. We see this. Now, if you were going to think about interferon, if you were in charge of the immune system and you were thinking about interferon, the cells that you would want to have make interferon were any cell that could be infected with a virus. Turns out that that's pretty much every cell of your body. So every cell of your body could make interferon if it wanted to. Some are particularly good, some are particularly bad. Except for red blood cells, because red blood cells don't have a nucleus. And so because they don't have a nucleus, they're useless to viruses. So they don't do any of this stuff. But all of our nucleated cells, they make type 1 interferon, interferon alpha, and interferon beta. And interferon alpha and interferon beta bind to a receptor. That receptor is very helpfully called the type 1 interferon receptor. I usually refer to it as IFNAR. which is the interferon alpha beta receptor. So we've got this receptor that binds to interferon. Note that this cell that we're seeing here is not the cell that got infected. This is some other cell. It's the neighbor. It's receiving interferon from the infected cell. When this cell receives interferon, this cell will now start to make a response. It will start to transcribe new genes. And those new genes will put this cell into an antiviral state. So here's our original infected cell. It's a virus production factory. It's a goner. We're not doing anything good for that cell. Maybe, as Andrew suggests, it will undergo apoptosis. But that cell sends out a message, interferon. And that cell says to its neighbors, hey neighbor, prep for a virus coming. Get ready to not let viruses reproduce so that you don't have to die too. So it is only me that has to die. And so this cell now goes into the antiviral state. And so if this cell gets infected, or if a virus tries to infect this cell will not allow viral replication. So this cell is in the antiviral state. So note that the antiviral state is not in the cell that got infected. It's in the neighbor. It's happening in a cell with the interferon receptor because of receiving interferon made by the infected cell. When type 1 so here you can see a virus, we're making type 1 interferon through that PRR signaling process. Here's my type 1 interferon. It's signaling to my neighboring cell. See, I made a line so you know it was a neighboring cell. And this might lead to thousands of different proteins being made after transcription. It's a whole list of proteins. They're generally known as the ISGs, or the interferon stimulated genes. Most of them have a promoter that's in common. And what's probably important is the combination of which ISGs are made. When you look at the paper for Monday, the fourth figure is going to be looking at all these different genes that are transcribed. Really, it's looking at which ISG combos are made. And so that's kind of what you're thinking about in that's to me, that's quite possibly the most important figure. And it's really thinking about which ISGs are made after infection. And so the combo is probably important, but that's something we're still not totally clear about. And what we need to talk about that we don't really have time for right now is how exactly these ISGs block infection. How do they make the cell antiviral? So my guess is that does kind of tie into the paper on Monday, so we'll talk about it at the end on Monday. But I want you to think about, or a piece to be aware of is, sometimes when people hear about this, they're like, this sounds like a terrible system. And the reason why this sounds like a terrible system to me is that I would like my cells to be antiviral all the time. I don't want the viruses ever. Why are my cells not in the antiviral state sometimes? Like, you just told us that interferon comes around, it turns our cells into the antiviral state and they resist infection. Well, I would like my cells to resist infection all the time, please. And so we're going to have to talk about why we don't do that and what the problem with that is, which is related to the mechanism of how exactly they work. So we will talk about that next time after we talk about the paper. Have a great weekend. I'll see you on Monday.